So our next speaker is Kasper Krestensen from Maastricht University.
All right. So yeah, I just want to thank the organizers first for this great meeting. So I'm going to be presenting a recent project that we did at M4I in collaboration with Ambergen and Bruker, which we've titled MALDI-IHC-Guided in-depth spatial proteomics, targeted and untargeted MSI combined. So that's a bit of a mouthful, so I'm going to take you through it now, step by step. The technique that we use here in the lab is mass spec imaging, which was described previously by Eva. So I need you to remember a bit the basics of that for this talk. But basically what we can do there is do untargeted MALDI imaging of lipids or metabolites in the tissue, and then we get a lot of untargeted images of our tissue of interest. Now, what Ambergen has developed is this method of MALDI-immunohistochemistry, where we combine MALDI imaging with these photocleavable mass tag antibodies, which are antibodies linked to an ionizable mass tag here, as well as a fluorescent tag. And then when you stain your tissue with these antibodies and release the mass tags just with UV radiation, you can do targeted imaging instead of untargeted imaging, because you image the released mass tags instead of the actual targets in the tissue. So that allows us to do really nice multiplexing images here, which sort of goes around the standard issues that you would get with just fluorescence imaging regarding wavelengths overlapping. So our workflow is just a standard mass spec imaging workflow. However, what we wanted to do is incorporate the spatial omics that we do. So we want to cut out regions of interest based on our MALDI-immunohistochemistry imaging, and then do triptych digesting and proteomics sort of the regions of interest, and see what we get out of that in comparison with the images that we also have. So we did that for for a number of different breast cancer tissues that we had, and as we can see here in the spectrum up here, we got an antibody panel of 6 different targets. So we have CD20, HER2, actin, vimentin, CD68, as well as pan-cytokeratin. And what we were quite happy with in the first case was that we were able to detect all of our target molecules here. So we were able to detect all 6 quite effectively. But what we could also do, or what we were also happy with, is when we look at the images that we obtained, is that each of the different probes, so to say, have a differential spatial distribution. So if we take a look at, for example, CD20, which is a marker for B cells, we see a very localized abundance of signal up here. And when we compare that to the fluorescence staining, which is also possible due to the fluorescent tag on the probes, we can see that it matches very nicely with this yellow target up here, which is the CD20 staining. Yeah, what we also see is the actin very nicely co-localizing around the vessels in the tissue. So everything is sort of making sense with what we see in our images. Moving on to sort of the proteomics part of this, the spatial omics. So this is a bit of an overview of what we do in the spatial omics when we're talking mass spec imaging. So we first, of course, take an optical scan of our tissue, and then we do MALDI-immunohistochemistry in this case. Then, based on the images that we get, we do a computational clustering just based on the abundance of the different ions that we see, and then cut out the regions, as Eva also mentioned, with the laser microdissection device, which allows us to collect regions and then extract the proteins or lipids or whatever we want from a more specific region rather than homogenizing the whole tissue. So in this case, we did a triptych digest of the regions. and then sort of looked at which sort of pathways and proteins we saw in this. And what we saw, what we got out of that mainly was 3 different clusters. So we have the first cluster here, the teal cluster, which was mainly correlating with the localization of pan-side keratin, and looking at the specific pathways and overrepresented proteins that we saw in these pathways. We saw sort of pathways that we would expect from cancer tissues. So a lot of DNA repair, DNA replication, and Rho GTPase signaling, which is nice to see when we have cancer tissue, of course. So for the second cluster, the yellow cluster here, it was mainly, so this cluster was correlating with the spatial localization of our Vimentin and CD20 marker. And what we actually see here is that there is a overrepresentation of immune system pathways and proteins, which makes sense if we go back and look at which marker we're looking at here. So CD20 is a marker for B cells. So that we see an overrepresentation of immune cells makes a lot of sense in this case, and sort of reinforces the idea that we do, or shows how the specific targeted proteomics here shows the upregulation of the immune system here. And finally, if we look at the last cluster, so the blue cluster was correlating mostly with the actin. And what I want to highlight here is we saw an upregulation of vesicle-mediated transport, which in the context of tumor cells and actin can be maybe correlating with the metastasis that we see. So the tumor cells sort of invade out into nearby tissue, and by doing that, they really use these actin-filled protrusions from the cells. So all in all, we sort of see the importance of, or the sort of, the importance of considering intratumor heterogeneity here, in that we see differential pathways upregulated in different parts of the tumor tissue. So we sort of compared that with the other option that we have for proteomics in mass spec imaging, which is the on-tissue digestion, which is sort of an untargeted bottom-up approach that we do. So we spray trypsin on just our whole tissue, and then we measure the tissue itself, and then we get the spatial distributions of the peptides that we get based on the digestion. So what you can see from this spectrum is that it's very, very messy, and it actually is also one of the downfalls of this method, is that it's very hard to extract specific proteins and peptides from this. But still, we were able to extract a lot of the different peptides that we also saw from the LC-MS data previously. So the keratins, the actins, the Vimentin. But what we also saw was something like fibrinogen alpha and fibrinogen beta, which have previously also been related to cancer tissues. And here we see very specific localizations again, which is interesting because these we actually did not see in the LC-MS data, and then that we saw them here sort of shows the importance of looking at the whole tissue at once. So comparing the two, we saw a very nice overlap overall. However, of course, we saw a lot more peaks in the LC-MS data compared to the MALDI data, but still that they both sort of represented or showed the importance of considering intratumor heterogeneity was a nice sort of result for us to end on. So finally, I just want to thank all the people at M4I that was involved in this project, as well as the people from Ambergen and Bruker for sponsoring the pitch, and the organizers for a nice day today. Thank you.